All right. Good afternoon, everyone. Welcome to the SDB Ethel Browne Harvey Postdoctoral Seminar Series. I am Sudha Rajderkar, transitioning from my postdoc from the Lawrence Berkeley National Laboratory. I will be moderating the sessions today with Melissa Bentley-Ford from Emory University. She is SDB's graduate student trainee representative. We are excited to highlight the research of our excellent postdoctoral members. Today's speakers are Jennifer McKey and Frank Macabenta. Each speaker will have about 20 minutes for their presentation, followed by a 10-minute Q&A. Jennifer McKey is a postdoctoral associate in the Capel Lab at the Duke University Medical Center. She is interested in understanding mechanisms of ovarian development using the mouse as a model system. Jennifer completed her graduate studies in France at the University of Montpellier. She has presented her research at several invited talks and meetings, including the SDB. Jennifer has received numerous awards throughout her career, including the American Cancer Society Postdoctoral Fellowship, and most recently, the NIH K99 Pathway to Independence Award to continue research in reproductive biology. Today, she will tell us about the developmental dynamics of ovarian morphogenesis. I request the audience to type in their questions in the Q&A box, and Jennifer will address those at the end of her talk. So off to you, Jennifer.
Hi, everyone, and thank you, Sudha, for that kind introduction. I'm really thrilled to be a part of this incredible seminar series, and I'm excited to share with you today my recent efforts to better understand morphogenesis of the mouse ovary. During fetal development of the mammalian female, the ovary first arises as an elongated population of cells growing on top of the mesonephros. And among these cells, supporting cells differentiate into granulosa cells, while germ cells differentiate into oocytes. And as development of the ovary continues, these two cell types assemble into individual structures called ovarian follicles that are defined by the presence of one oocyte and 2 to 3 granulosa cells. And as importantly, the follicles that are assembled in the middle of the ovary in this region called the medulla will initiate growth shortly after birth, but they'll all be depleted by the onset of puberty and do not contribute to fertility past the first litter. In the meantime, oocytes that find themselves closer to the surface of the ovary in this region called the cortex become supported by newly differentiated granulosa cells in the perinatal phase, during late gestation, and assemble into primordial follicles during the perinatal phase. But these will remain quiescent throughout juvenile development and will only begin to activate after puberty in a cyclic fashion. This pool of cortical follicles is never replenished, and so it is responsible for the entire reproductive lifespan of the adult female in mammals, at least. And this really highlights how important it is for us to better understand ovary development so that we can hopefully harness these findings in the clinic to address certain cases of female subfertility. And while our knowledge of the molecular pathways that are involved in the differentiation of individual ovarian cell types is increasing, we still have a poor understanding of how structural events structural rearrangements, of the ovary as a whole might be contributing to this regionalization and thus to the determination of female fertility. And we already knew from studies on testis morphogenesis that functional differentiation of the gonad is tightly linked to structural changes. And the stepwise morphogenesis of the testis had already been extensively studied. However, the morphogenesis of the ovary has been much harder to access, mostly because the architecture of the ovary is not as well defined as that of the testis. And so we really didn't have the right landmarks and the right approaches to really visualize how this architecture is established. And so I chose to tackle this question during my postdoc, and I've spent the better part of my time in the CAPA lab trying to establish this pipeline for investigating ovary morphogenesis. And so to do this, I developed and implemented a clearing approach that combines idisco tissue clearing and cubic clearing. And with these methods, we can take whole ovaries that were dissected kind of in their native conformation and context and render these fully transparent to image with the light sheet microscope. And here you'll see an example in this video of an E14.5 mouse ovary that was labeled with VOXL2 to visualize the ovarian domain and PAX8 to visualize the reproductive tract. And using MRS software, we perform isosurface segmentation so that we can generate 3D models based on these stains. And then we can spin these 3D models in every other direction and kind of look at the ovary from every angle and better appreciate the changes in the context and the conformation of the ovary as it develops. And so the first hallmark of ovary morphogenesis, and probably the most obvious one, is that the ovary transitions from an elongated to a crescent-shaped domain during late gestation. And this is illustrated here with both traditional confocal microscopy images and also the 3D models based on our light sheet images. And what we found is that the anterior and posterior poles of the ovary rise towards the medial axis of the ovary. And this is associated with an inflection of the medial part of the ovary that leads to an increase in the concavity of the lateral face. And these changes are suggestive of a folding mechanism. And then, as I mentioned, the regionalization of the ovary into cortical and medullary regions seems to be functionally significant. And so I was interested to see how the folding of the ovary might correlate with this regionalization. To investigate this, we used the LGR5 GFP reporter line, in which the ovarian surface epithelium is labeled with GFP, and this serves as a proxy for the formation of the cortex. Early during development, before the ovary folds, the ovarian surface epithelium is only visible from the ventral lateral side. But as the ovary folds and it looks like the ovarian surface epithelium wraps around towards the dorsal side of the ovary, and everything that was initially found on the dorsal side of the ovary now seems to ingress and be engulfed inside of this folded ovary. And we believe that these transitions lead to this functional regionalization between cortical and medullary domains. So we then became interested in investigating the correlation between morphogenesis of the ovary and the developmental dynamics of proximate tissues, starting with the Müllerian duct, which is the female reproductive tract, and gives rise during development to part of the vagina, the uterus, and the oviduct, which is the segment most closely associated with the ovary. Early during development, the oviduct is found on the dorsal lateral aspect of the ovary. But really quickly, we see that the oviduct translocates across the lateral face of the ovary. And by birth, the oviduct is fully coiled, and these coils are all found on the ventral aspect of the ovary. If we look at this in a little more detail and focus our attention on this region here labeled with the yellow asterisk, which is the infundibulum of the oviduct, we found that the translocation of the long segment of the oviduct led to a relocation of the infundibulum from the anterior pole of the ovary to the medial edge of the folded ovary. And as the oviduct expands and grows around the ovary, it leaves behind it this growth of mesonephric tissue that fully encapsulates the ovary by birth, where all of the coils of the oviduct form the ventral rim of this ovarian capsule. So these images really highlight a close association between ovary folding, encapsulation, and also oviduct development. So we then became interested in investigating mechanical components that could be affecting change in both the ovary and the oviduct simultaneously. The position of the ovary in the body is maintained in part by the cranial suspensor ligament, which tethers the ovary to the dorsal abdominal body wall. And here you can see a urogenital complex from an E14.5 mouse line where both the gonad and the adrenal are labeled with GFP. And we're able to label the cranial suspensory ligament with antibodies against tensin C. And here you can see this ligament exit the anterior pole of the ovary and line the entire lateral side of the kidney all the way up to the adrenal, where it plugs into the body wall behind the adrenal. And this link is maintained throughout development, where in a neonatal urogenital complex, we still see this cranial suspensory ligament line the entire lateral edge of the kidney between the ovary and the adrenal. And so one possibility is that the growth of the kidney between these two organs leads or exerts mechanical tension on the cranial suspensory ligament, which could be driving ovary folding. So we looked at this in a little more detail here on isolated ovary mesonephros complexes, and we found that the cranial suspensory ligament lines the entire medial edge of the ovary and seems to be attached here near the anterior pole, and also seems to link back to the infundibulum of the oviduct. And after the ovary folds, the cranial suspensory ligament is relocated and now kind of runs across the entire medulla of the ovary. And this relocation was consistent with an attachment of the cranial suspensory ligament to the original anterior region of the ovary and to the infundibulum of the oviduct. And again, just looking at the same thing here with a different marker, RUNX1, which also labels the cranial suspensory ligament, we see this discrete population of cells at the medial edge of the ovary, linking directly back to the infundibulum before continuing its trajectory over to the body wall. And again, this link was remained throughout development. So this really shows that tension on the cranial suspensory ligament could be driving both the relocation of the infundibulum and also the folding of the ovary. So in our investigation of proximal tissues and their relation to ovary morphogenesis, we also investigated the rete ovarii, which is an epithelial network that is strongly associated with the ovary. And what is kind of crazy is that the rete ovarii has been described in really old literature and as this, quite significant structure that develops with the ovary, but it's been sort of forgotten in more recent textbooks. And we rediscovered this structure using antibodies against PAX8, and we've been really interested in investigating its development, but also finally addressing its function during ovary development and also in ovarian functions. But for the context of this study, we really investigated the correlation between the development of the rete and the morphogenesis of the ovary. The rete is divided into 3 different regions: the intra-ovarian rete, which is embedded inside of the ovarian domain, the extra-ovarian rete, which develops outside of the ovary, and then the connecting rete, which links these 2 regions together. And when we look at these in the context of ovary morphogenesis, we found some interesting correlations where the extra-ovarian rete truly expands within the ovarian capsule as the ovary becomes encapsulated. The intra-ovarian rete always remains firmly embedded inside of the ovarian domain, and the shape of the intra-ovarian rete actually follows the shape of the ovary as it folds. The connecting rete always resounds at the boundary between the ovary and the mesonephros, and one hypothesis is that the connecting rete might serve as an anchor of the ovary to the mesonephros and kind of keep the ovary in place as it's undergoing all of these structural changes. So having observed these correlations between the development of the ovary and the development of the oviduct and rete ovarii, we then became really interested in addressing whether or not these tissues might drive ovary morphogenesis. And so to investigate this, we initiated a collaboration with Richard Berenger at MD Anderson, who had a model for disruption of the Müllerian duct that also presented with disruptions in the rete ovarii. And these are embryos that carry different combinations of PAX2 and PAX8 deletion alleles. And so with these models, I was really interested to address whether expansion of the Müllerian duct truly drives ovary encapsulation, whether tethering of the ovary to the infundibulum is what drives folding, and then finally, whether the rete ovarii is even involved in ovary morphogenesis. So I performed a study on an entire allelic series with these mice, but for the sake of time, I'm only going to show you the genotype that led to the most striking phenotype, which occurred in PAX2 homozygous deletion, PAX8 heterozygous deletion animals. So in a control animal, you can see that the oviduct has expanded, and we see this long segment of the oviduct wrapped around the ovary. However, in our mutant samples, we found that at E17.5, this elongated segment of oviduct was absent. And interestingly, these ovaries were also not encapsulated. So we believe that this shows that the encapsulation of the ovary does rely on the presence of this elongated segment of the oviduct. However, at first glance, it doesn't really look like this ovary had any other disruption in morphogenesis, and it seems to have folded normally. But I do want to draw your attention to the fact that we have this small fragment of tissue that remains here, and we believe that this is the infundibulum that resisted the deletion of PAX2 and PAX8. And when we looked at the cranial suspensory ligament in these animals, we found that this small piece of infundibulum was actually sufficient to tether the ovary to the cranial suspensory ligament, which could explain why these ovaries still folded. So to actually be able to address this question of whether tethering of the ovary to the infundibulum drives folding, we now need to develop more or better models where the entire oviduct, including the infundibulum, are depleted, and hopefully this will lead to phenotype and folding. And I'm currently working on developing these models. As I mentioned, these animals also presented with disruptions in the radiovarii, and indeed you can see here in the sample that all that remains is the connecting radi. Both the intraovarian radi and the extraovarian radi are depleted in this animal at E17.5. Again, this ovary seems to have folded rather normally, and so at first we were a little disappointed and thought, well, maybe this is telling us that the radi really isn't all that important for ovary morphogenesis. But I've now started looking at earlier stages with these alleles and found that before the ovary folds, the radi, it does not seem that as disrupted as it does here. And so what's likely happening in these animals is that the radi initially develops but degenerates by E17.5. And so if we really want to address a role for the radi in ovary folding, then we again need to develop better models where depletion of the radiovarii occurs before initiation of folding. And I'm also working on establishing those currently. So having defined these hallmarks of ovary morphogenesis and their relationship to the development of tissues associated with the ovary, I'm now really looking forward to moving forward with this project and to integrate these processes of ovary morphogenesis with the events of ovary folliculogenesis and functional differentiation of the ovary. And this is going to be an important first step towards my long-term goal, which is to really understand how these structural transitions that occur during fetal life are how these occur and how they contribute to the establishment of female fertility and thus act as a determinant of reproductive longevity in the female. And I'm hoping to address these questions by implementing a multi-scale approach that will investigate how interactions between different cell subtypes within the ovary drive the allocation of follicles into cortex versus medullary regions. As I mentioned a lot today, I'm really interested in finding out how the ovary folds and whether this is through extrinsic forces like the cranial suspensory ligament, or if instead it's through intrinsic rearrangements of the ovarian domain. And then once I figure out how the ovary folds, I really want to see if I can manipulate this event so that I can understand how folding of the ovary might contribute to this functional regionalization. Finally, I am really interested in investigating tissue interactions, their nature, and also their role in driving ovary morphogenesis, folliculogenesis, and adult fertility. And so one example that I mentioned today is investigating the interaction between the rete and ovarian cells and understanding how that might contribute to establishment of architecture and function of the ovary. And so with that, I want to thank all of the members of the CAPEL Lab for providing such an inspiring and fun workplace. I am just so grateful to Blanche for her continued support and encouragements. I am really thankful to Richard and Ale, our collaborators who helped us with the PAX2, PAX8 mice. I couldn't have done any of this work without the helpful staff at the Duke Light Microscopy Core Facility, Ben and Lisa. I'm also really grateful for all of our sources of funding. You can reach out to me on Twitter or by email. You can also read the full story if you're interested. And I once again want to thank the organizers of this incredible seminar for giving me the opportunity to share my work today. And thank you all for your attention. I'll be happy to take any questions.
Thank you. That was an excellent talk. I have a question from Chris Wright here, asking, is it known at all if this is a very similar or different in human or primate development?
Yeah, so that's a very intriguing question and one that keeps me up at night. The anatomy of the reproductive system in humans is different. And in women, for example, the ovary doesn't seem to be encapsulated, at least not in adulthood. It's unclear whether it has a capsule during development that just degenerates later. In terms of whether it folds and those kind of changes in shape, it's really unclear because a lot of the fetal work that's been done on human ovaries is either really early or we have kind of like the later stages post-birth. And so it's hard to tell whether the mechanisms are similar, but that's something that I'd really like to know and hope that I can engage in some collaborations that will help me find that out.
Thank you. I want to ask a follow-up to that. So in terms of the ovarian reserves, is there a preferred strain of mouse that you use to do these experiments?
So the data that I showed today is mostly on a cross between Black6 and CD1 or pure CD1. We just use those because they have really great consistently large litters. I do know that different strains of mice have different ovarian reserves, and I think that's also something that's really that I would really want to investigate, is to see whether morphogenesis of the ovary changes slightly in the different strains and kind of do that comparative approach, but I haven't done that yet.
Thank you. There's a question from L. Robertson. She says that she thinks the oviduct finished coiling postnatally by P15, and she's curious if the ovary continues to undergo morphogenesis postnatally, and if so, does it contribute to or help organize oviduct coiling?
Oh, that's a really good, I hadn't thought of it in that sense. That's a really good question. Hopefully I can manipulate this someday and be able to address that question functionally. In terms of whether the ovary still undergoes morphogenesis postnatally, I'd say that because we have this wave of activation that happens shortly after birth, the size of the ovary really changes a lot postnatally because all these growing follicles obviously take a lot of room. And there's also a lot of kind of extrinsic components that are still arriving into the ovary postnatally. So there are some structural rearrangements that occur during the perinatal phase. But in terms of whether the shape really changes, I don't think we can say that, except for like this just enlargement that comes with the growth of the follicles. But yeah, I think that's a really good point. And I might add some 3D models for those late stages and see whether oviduct coiling changes something at that point.
Thanks. Chris Wright has another question saying, there's a paper from 1974 in Nature on the rete ovari as a potential trigger of meiosis. Do you have, what are your views on that?
Yeah, I think that's a compelling hypothesis. And again, it's something that I'd really like to find out. So it is true in that paper, they suggested that that the rete could be a trigger for meiosis. This is based on the fact that we think that there's something in the anterior region of the mesonephros that makes retinoic acid that triggers female meiosis, and obviously the rete is what's there in that anterior region of the mesonephros. So I think it's a really compelling hypothesis, and I think meiosis is definitely one of the first things that I'll look at once I have that good model for ablation of the rete. But so far I haven't been able to ablate it early enough to actually investigate that. And I don't think in the literature it hasn't been functionally tested. It's only been suggested.
Okay. We can have maybe another question if anybody has any for Jennifer. In the meanwhile, I might ask one. So in terms of birth defects, are there known conditions in humans, where there are congenital defects reported with changes in the rete?
There might be. The rete is not something I think that physicians would think to look at, just because it's really true. If you look at the anatomy and physiology textbooks currently, the rete is maybe a one-liner if it's a really good, thorough textbook, but for the most part, it doesn't appear on the diagrams. And so I think it's just something that we haven't thought to look at really, but it could be, and if it were, then that would be really interesting to find out, and to find out, and to help with kind of the functional study of the rete. It's something that's really hard to see, even in the mouse. In the adults, in the adult mouse, the rete is embedded inside of this fat pad, and so if you don't have a marker to see it, we're fortunate that we have like a GFP reporter line, so we can actually dissect it with fluorescence. But if you don't have that, it's impossible to see it. So I'm not blaming anyone for not looking at it. It's really easy to miss, and we got really fortunate using these PAX8 antibodies, and we're able to label it that way.
Okay, on to our well, there's one more comment from Chris, which he says, lots of cysts or metaplasia noted in rete in some species, and this could be a predisposed tissue for cancer.
Yeah, I agree. There's a lot of cases in dogs and cats where there's cystic development of the rete, and I agree that it could be an origin of ovarian cancer, and that's also something that I'd love to investigate.
Well, thank you so much, Jennifer.
Thank you.
We wish you good luck with your research, and on to our next great speaker. Thank you.
Thank you so much for that great talk. My name is Melissa, and I am currently the graduate student trainee representative for SDB. And it is my pleasure to introduce our next speaker today, Dr. Frank Macabenta, who is currently a postdoctoral fellow in the Stathopoulos lab at Caltech. And today he is going to be talking to us about his work that he is doing in that lab. And without further ado, I am going to turn it over to you, Frank.
All right. Thank you very much for that introduction. Good afternoon, everyone. I'm excited to share with you an upcoming story coming out of our lab, talking about a quality control mechanism that we identified via the intersection of two different signaling pathways. And a way I like to think of this is a tale of love, death, receptors, and zombies, with apologies to Tom O'Donnell. So as developmental biologists, I think all of us could appreciate that in organ development or in the formation of higher order biological structures, there is, in order to acquire a very specific shape, there's this coordinated regulation of several different parameters, one of which is cells, specialized cells undergoing cell shape changes to allow for specific functions. For instance, you've got neurons who are able to innervate distant tissues. And you also have to have patterns of cell proliferation, which account for differences in wing shape in different insect species, for instance, as well as coordinated cell movement, such as invagination, allowing for branching morphogenesis in developing lungs. And finally, you also have this coordination of apoptosis or programmed cell death, which allows for this highly stereotyped pattern in a lace plant leaf. And really, the dysregulation of any of these parameters can lead to severe morphogenetic disorders or disruptions in function and disease. And one mechanism that really encapsulates this coordinated regulation quite elegantly is cell competition. Now, cell competition is a way for a collective of cells to eliminate hypoplastic or abnormal cells while allowing for a steady state or homeostasis to be maintained via compensatory proliferation. And as shown in this example over here, where the abnormal cell is highlighted in red, there's extrusion that happens, but there's an elegant choreography of remodeling of stable junctions in order to be able to allow for compensatory proliferation to take place and seal up that gap. Now, epithelial cells, epithelial cell collectives that have stable junctions have really been the paradigm for collective migration. But we now know that there is a continuum of cell states that exists in various forms of development. Anywhere between the epithelial to mesenchymal states, you get also this variety in the number of cells that have stable adhesions between neighboring cells. And so that really leads us to the question of whether these more transient adhesive contacts allow for quality control as well. And to investigate this question, I used the genetically tractable Drosophila fruit fly system and specifically look at a population of collectively migrating muscle precursor cells called caudal visceral mesoderm cells. Now, these cells undergo synchronous bilateral migration along a trunk visceral mesoderm tract, as highlighted here in this diagram by cyan color, before eventually undergoing myoblast fusion to form the larval midgut musculature. So the CVM cells seed the longitudinal fibers in this muscle structure, while the TVM seeds the circular muscle fibers. And as you can see from this phalloidin staining, you can actually track any disruptions in this highly stereotyped pattern. And something we could also do is visualize this process live. This is an embryo with a CVM-specific GAL4 construct expressing 6XM cherry. And what this does is it allows us to visualize different events over the course of migration, including proliferation. And something we observed are wandering cells that seemingly leave the cohort and undergo cell death. Now, the cell death is a specialized form of cell death called anoikis. And this anoikis cell death is defined as the adhesion from an extracellular matrix substrate. Now, anoikis is really relevant to public health because the acquisition of anoikis resistant is a precursor to various types of metastatic cancer. Now, segueing back to the CVM, previous research conducted in our lab identified a critical role for FGF signaling in controlling the coordinated movement and survival of CVM cells. The FGF receptor encoded by the gene heartless is expressed in the CVM as visualized by this reporter for the CVM overlaid with mRNA probe against heartless. while the FGF ligands Pyramis and Thisbe are expressed in the TVM track. And something that I hope you can appreciate is that the mutation of these FGF signaling components results in these gross migration defects over the course of CVM migration. As you can see from these movie stills on the right, mutations against the FGF receptor heartless or taking out both the Pyramis and Thisbe ligands results in merging and inappropriate migration of the CVM cells. Something we could also look at is cell death over the course of CVM migration, and it seems to proceed even in wild type. Using a tunnel assay, which is the green signal here, you can see that there is a small population of cells that does undergo cell death, even in wild type. However, when you look at mutants for the heartless FGF receptor, you see this really wholesale loss of cells, such that nearly all CVM cells undergo apoptosis. And so this led me to wonder how this temporal regulation of cell death in CVM cells is accomplished. Because if you recall, the CVM cells are able to undergo migration early, but it's only after they round this posterior turn in the embryo that they undergo this wholesale cell death. And to summarize what we know so far, we know that the heartless FGF receptor is expressed in the CVM and that it interacts with FGF ligands expressed in the TVM. Now, research conducted in our laboratory has also found a link between FGF signaling and the promotion of integrin-mediated adhesion to the TVM substrate, as well as survival. And to investigate these questions further, I used an antibody against teramera or Te, which allows me to label the CVM nuclei. And Te is actually a really useful metric for assessing cell survival because, as you can see, when looking at an embryo that has been stained against Te, as well as this GFP reporter that allows us to visualize the CVM cell membranes, Te signal is absent in cells that have extruded from the rest of the CVM cohort and undergo cell death. And so if you compare the wild type versus heartless images below, the Te signal is almost largely absent in the heartless FGF receptor mutant. An additional thing we observed is the expression of this cell death protein called head involution defective, or HID, in the CVM over the course of migration. Now, HID is an ortholog of mammalian SMAC Diablo. And what I found striking about HID expression is that it seems to be temporally regulated. In the top panel here at stage 11, when the CVM are still migrating towards the posterior turn, HID expression is largely absent. However, by the time the cells reach the posterior turn, you start to see HID expression ramp up and perdure through stage 13. And this seems to be correlated with the timing of cell division in these migratory CVM cells. And to visualize cell division, we used an antibody against phosphohistone, which is shown here in white. And so to investigate a potential link between HID mRNA expression and cell division, I looked at mutants for the gene string, which encodes a CDC25 phosphatase that acts as a universal mitotic activator in Drosophila. And so what I observed is that HID mRNA expression is delayed significantly in string mutants, such that there is very little to no signal by the time the cells have rounded this posterior turn, but eventually it does start to ramp up at stage 13. So taking what we know so far, we decided to count the number of CVM cell nuclei using Te antibody staining to compare various mutant backgrounds. So I mentioned that heartless mutants have a near total loss of CVM cells by stage 13. When we generated double mutants for heartless and HID, we found that it attenuated the cell death defect observed in heartless mutants. And a similar phenotype was observed when we generated double mutants for heartless and string. As you can see here in this graph, showing the number of cells in the y-axis and the genotype in the x-axis. And so this led us to tenuously propose a link between string dynamics and the onset of hit expression in these migratory CVM. And we decided to follow up on what actually regulates string dynamics in the CVM over the course of their migration. And to do this, we looked to ChIP-seq data as well as enhancer data and identified a visceral mesoderm specific enhancer for string. And so we generated reporter constructs that show us that this enhancer is active in both the CVM and the TVM. And an interesting observation we made by looking at PMAD ChIP-seq data is that there seems to be strong occupancy for PMAD in this enhancer. And PMAD is a downstream transducer of BMP signaling. And so the next thing we wanted to see is, where are these BMP signaling components? And one of the first things I looked at is the expression of the BMP ligand decapentaplegic, or DPP, which is a very broadly expressed morphogen throughout Drosophila embryogenesis. And what you can see from these HCR stainings, in addition to TAE antibody signal, is that DPP is expressed in this bolus, parasegment 7 of the TVM, as well as in the ectoderm, as well as what would eventually become the cardiogenic lineage. Additionally, we looked at RNA-seq data that we previously published that features genes that are enriched in the CVM. And we identified this gene encoded by TOC that supports expression of this metalloprotease that is secreted and promotes BMP signaling by cleaving short gastrulation, which is an inhibitor of DPP signaling, mediated signaling. So what TOC does is it cleaves SOG, frees DPP ligand for downstream binding to thick veins receptor, thus allowing BMP signaling to occur. And a lot of the initial work that was done characterizing TOC was performed by this talented undergraduate student named Miriam, who is now currently a grad student at Stanford. And so what we next wanted to do is investigate how these various mutant backgrounds contribute to CVM cell behavior over the course of migration. So what we first did was we looked at dorsal view embryos and counted the number of TAE-positive nuclei and compared them before the CVM cells reached the posterior term and after at stage 13. And to summarize, what you can see in the left-hand panels is that between wild-type heartless TOC mutants, TOC heartless double mutants, and BMP receptor and heartless double mutants, there really isn't a significant difference in the number of CVM cells. However, after the cells have rounded the posterior turn, we start to see differing trends occur. In TOC single mutants, we found that there is a reduction in the number of CVM cells when compared to wild type. But when we generated double mutants for TOC and FGF receptor heartless, we found that there was an attenuation of the cell death phenotype, resulting in a rescue of the number of CVM cells over heartless mutants. And a similar effect was observed when we generated double mutants for the thick veins BMP receptor and heartless, which led us to uncover a parallel signaling pathway that seems to contribute to this mechanism for controlling the onset of hit expression in migratory CVM. So CVM expressed TOC cleaves SOG inhibitor, thus freeing DPP ligand for activation of the thick veins receptor, which in turn allows for activation of the string enhancer to control cell division dynamics to promote hit expression. And we wanted to investigate the nature of this process further. And to do this, we used this fly fluorescent ubiquitin associated cell cycle indicator, or FlyFucci, which allows us to visually track the stages of the cell cycle over the course of migration. Now, Fucci takes advantage of this property of cell cycle proteins, where each protein is degraded in a ubiquitin-dependent manner prior to the onset of the next cycle of the cell cycle, which allows for this unidirectional nature of the cell cycle. And in this case, we have the degrons of the E2F1 transcription factor and cyclin B conjugated to GFP and MRFP. So to track where you are in the cell cycle over the course of CVM migration, you only need look at the color. So CVM cells that are red are in S phase. And as the cells transition into G2, you start to see that the cells adopt a more yellow color due to the overlapping red and green. And as they transition past M phase and into G1 phase, you start to see only green signal appear. Now, if we lay these images out in succession, you can actually see a nice overview of the transitions of the various cell cycle stages over the course of CVM cell migration. And when we look at a heartless mutant, you can see that it really follows the pattern of cell cycle progression as in wild type, albeit the loss of the CVM cells at the conclusion of migration at stage 13, which suggests that in heartless mutants, these cells are able to undergo mitosis, but die shortly after reaching G1. However, when we expressed Fuchi in a heartless Toke double mutant background, we find that the cells are able to transition into G2, but become arrested. As you can see throughout stage 12 to stage 13, there's perdurance of this yellow-orange signal, indicating cell cycle arrest. And this led us to think that we can perhaps manipulate this mechanism for inducing cell death in CVM cells over the course of migration. And to do this, we used the Gal4 UAS system to express either a dsRNA construct against the mitotic activator string or express string protein in the CVM cells in a heartless mutant background. And what we found is that expressing string dsRNA in the CVM recapitulates the heartless string double mutant data that I showed earlier, where you have survival of the CVM cells at the conclusion of stage 13. However, the opposite effect is observed when you express string protein, specifically in the CVM, even in a Toke heartless double mutant background, which should have resulted in survival. This instead results in precocious cell death but in stage 11 prior to reaching the posterior turn, suggesting that BMP signaling's ability to promote cell division is necessary and sufficient for positioning the timing of cell death in CVM cells before they reach their destination. And so this leads us to the following model, where over the course of CVM migration, access to DPP signal allows the cells to undergo cell cycle progression. And whether they survive or not depends on their proximity to FGF ligand. And so we can see these different scenarios where if the cells have lost access to FGF ligand, they've lost this protective nature of FGF to antagonize HID cell death protein from promoting cell death because CVM cell cycle progression proceeds in the presence of secreted DPP ligand. Now, if we remove both FGF and BMP signaling components, we see that there's G2 arrest due to an inability for the CVM cells to move forward and undergo the transition from G2 to M and into G1. Now, when you look at these undead cells, you don't really think of zombies, but I can tell you that there is one thing that they seem to have in common with zombies, and that's an apparent affinity for brains. So if we look at mutants for heartless and HID, we find that there's inappropriate invasion of these CVM cells into the embryonic ventral nerve cord, or CNS, as visualized by this BP102 antibody staining, suggesting that it's critically important that these extraneous cells be removed lest they invade distant tissues. And so this concludes my talk, and I hope that I've convinced you that CVM cell death requires HID being expressed in a temporal manner, and that this temporal dynamics of HID expression is controlled by this string enhancer in order to establish peak HID levels over the course of migration, and that in turn, this enhancer is regulated by paracrine BMP signaling via DPP. And finally, this system of cell cycle progression eliminates CVM cells that have lost contact with FGF signal, allowing for the highly stereotyped patterning of the midgut musculature over the course of development. And with that, I'd like to thank the members of the Stathopoulos Lab, especially my PI, Angela Stathopoulos, as well as our funding sources from the NIH, from the NIGMS and NICHD, as well as a Caltech Baxter Divisional Fellowship. And if you'd like to speak about this research further, please feel free to email me or follow me on Twitter. Even if you want to give me suggestions on books to read, as long as it's not post-apocalyptic fiction, because I'm good on that end. So thank you.
Thank you so much, Frank, for that fantastic talk. So just another reminder, from here we have time for a couple questions, and just take the time to type them into the Q&A box, and from there I can read them. But we do have one question from Chris Wright. Do you think it would ever be possible to test in your wild type or heartless mutant cells if they're able to be rescued back from anoicus commitment? If there is anoicus commitment, is it understood well?
That's a very good question. We're only beginning to understand the conditions that result in anoicus in CVM cells. Right now, it seems like the most direct connection is with FGF signaling, but we anticipate that there might be additional factors that contribute to this anoicus commitment. It's possible, and I've observed this in live imaging movies that I took of the CVM. There are some cells that seemingly leave the cohort and then snap back. And so they wander away from the rest of the collective, but somehow like, oh, it looks like I'm away from the collective, let me go move back. So this suggests that there is some time frame in which the CVM cells are able to correct any discrepancies in migration. And keep in mind that these cells have very transient contacts, but it remains to be seen how much this contribution is mediated by these transient contacts and whether the neighboring CVM cells even play a role in bringing back any lost cells.
Thank you. Okay, we have another question from Michelle Starsgiano. Nice work. So if the undead cells without HID or heartless migrate toward the head, do you believe there are other guidance cues that separate from FGF signaling? Have you tried to block cell death by P35 or other means?
Absolutely. Thank you very much for that question. Yeah, we did anticipate that there must be some kind of additional cue that might even be CVM expressed that is also expressed in the CNS. There are a number of immunoglobulin type cell adhesion molecules that seem to be shared by the CNS and the CVM. So it's possible that they just got confused and followed this other signal because they wandered away. It's funny that you mentioned the P35 because that's something that we have done where we express this baculovirus protein. And what that does, for people who don't know, is that it blocks the downstream effects of cleaved caspase and promotes survival of cells. And I don't want to give too much away because I'm saving it for another story, but we think that there's a potential additional role for this system that does not necessarily directly relate to elimination of abnormal cells only, but there might be a patterning function as well. So we do see a rescue, not quite what we expected. It didn't completely rescue the patterning, for instance. So yeah, thank you for that question.
Okay. A question from Vanessa Eld. Lovely talk. With the zombie cells, did they take on a clear fate and become integrated into the nervous system?
That's another great question. We did not investigate that to completion. It's something that I'm investigating further for a follow-up study. I will say that these cells seem to be adopting a senescence-like state, because by looking at Fuji signal in later stages, the cells that have wandered away from the primary CVM cell cohort appear to have re-entered the cell cycle. And so it really remains to be seen what the nature of their future interactions with other tissues are, whether they become integrated in other tissues or not.
And then one last question. So great talk. What happened? Oh, sorry, this is from Mark Cowrie. Great talk. What happens to the undead cells long-term if they end up in distant tissues?
Yeah, I guess that's similar to the previous answer. The short answer is we don't know yet. We do expect that they might be doing something, because I mentioned that Tolkien is a secreted metalloprotease, and we anticipate that there is possibly a non-autonomous effect by having these undead cells migrate to other tissues and settle there, because they could possibly be affecting the cleavage of SOG in other tissues, therefore affecting BMP signaling in these distant tissues. But these are all things that we're currently speculating on, but that's definitely something I want to follow further in the future.
And then the final, I think, which is more of a comment than a question, or maybe a suggestion from Chris, right, is the BORG Collective, B-O-R-G Collective. And so with that, I would just like to say thank you, Jennifer and Frank, for your excellent talks. This seminar has been recorded and will be available on the SDB website starting Tuesday, because Monday is a holiday. And please join us for next month's seminar on Friday, February 11th, where Joey Garci from Yale and Ellen Roberson from the University of Texas at Austin will present. And thank you all for coming.